Hi guys, welcome to Classification Processes Part 6. In the previous lesson, in Part 5, you would have gone through and understood that ecosystems are composed of a variety of habitats, including a microhabitat to ecoregions. You'd have also interpreted data to classify an ecosystem using things like SPEC's classification system, and you also would have had to explain how the process of classifying an ecosystem is important towards effective management, and you considered old growth forests, coral reefs, and also productive soils. We're changing gears a little bit here, and in this lesson you're going to be going through sampling. So by the end of this lesson, you're going to be able to do a few of these different things, and we're going to structure this over 4 lessons. It's quite a large topic, and particularly because sampling may be something you do for your student experiment, I'm going to give you that bit extra detail on that, so you're well prepared for either the external exam or your student experiment. So let's get going. So when we look at sampling, ecological sampling is a process of collecting data from a portion of an ecosystem and using that data to draw conclusions on the whole ecosystem. So if you want to find out something about that ecosystem, well, you do sampling to help you figure it out. Now, sampling is necessary because, for example, if I wanted to know how many organisms are here, if we're looking at caribou and their migration, how many are there? Well, I can't practically count every single one. So I do a variety of different sampling techniques to help me. Same if you go to a forest or on a sand dune. You cannot practically count every plant there. So you do different sampling techniques to help get an understanding of that environment. Now, sampling can be used for a variety of different things, and this is one of the first learning goals, its purpose. So the first thing is to estimate population size. So through different techniques, we can get an estimation of how many different organisms are in that area, okay? And that can be very important to look for conservation and a variety of different management strategies. Density, that's another thing sampling does. So density is the amount of organisms in a given sample area. So you can calculate how dense that population is, and you can also look at that distribution throughout that ecosystem. So is there zonation? Is there clearly more of this type of organism in this zone or more of another type of organism in another zone? So you can look at those distribution. Environmental gradients and profiles is another thing sampling does. So if you do sand dune or rocky shore, you can measure the different gradients across those zones. Zonation, probably a key one there. Again, looking at the sand dune, we've got those variety of different zones. So you've got your pioneer zone, that zone closest to the water. You then have your colonizer zone in the middle and your woodland zone in the end. So sampling can help us determine what organisms are found within each of those zones, and stratification, okay? So in this, stratification is breaking into different strata or areas, and we can have vertical or horizontal. So we're going to look at these different parts and hopefully get an understanding of how you do some of this sampling. So when looking at sampling, this is the steps you'd kind of go through to work through what you're going to do. So to start with, define precisely what is to be sampled and where. So think about what you might like to sample. Do you want to sample trees? Do you want to sample shrubs? Do you want to sample grasses? Are you sampling bugs or different mollusks on the intertidal zone? And then you need to figure out where that's going to be. So do you have a great site selection which enables you to conduct a large enough sample size? So that's the first thing you do. Then you determine the most appropriate sampling method. And we're going to teach you about all these different ones. So that might be random, systematic, or stratified sampling. And then the surveying technique to help you collect that data. So it could be a quadrat, a transect, a pitfall, or capture, mark, recapture, depending on the type of organisms you're looking at. Probably a lot of you will end up doing plants if you're doing sampling, just because they're quite easy, or you might do some of those organisms on that intertidal zone. After you've done that, you determine the sampling location. So you may have a site, and you might have picked the Sunshine Coast, okay? Then you might find your specific sampling location, which could be a particular dune on Bribie Island, or it might be, Point Cartwright if you're doing Rocky Shore, different things like that, and the sampling size. How large are you going to cover? So you go through those different steps, then you'll go out, collect data on either the abiotic and biotic factors in the sampling sites, depending on what your research question or what you're looking at. So let's start to look at some of those different sampling methods. We're going to cover those 3, so random, systematic, and stratified sampling. Let's begin with random. So random sampling is a method of selecting a sample in such a way that every possible sample that could be selected has an equal chance of being selected. This is very important that you ensure that your sampling is random. Now, you might set up a tape measure. It could be here, let's just say these are in meters. We'll try and be generous, so 20 by 20 meters of this zone, and you might do a random number generator. And the first random number that comes up is 10. So on that 10, you then go out and you mark that point. So this is where you're going to go from. And then on the other one, it comes up in random number generator, comes up as 8. So you find where 10 and 8 intercept there. So you'd simply go out. Now you might do this in a larger scale, 100 meters, and set up different things, or you may set up different grids. The key thing about random is that sampling is it takes out bias. You are not selecting a location, because it's very easy to go to a location and go, oh look, over there, that looks fantastic. That's where I'm going to put my quadrat. I've got many different species there. There's nothing much out there, and you're going to end up with bias and probably an overestimation of the population in that particular zone. So random sampling does help with that. So it does often involve using a table of random numbers from which you can select map coordinates or numbered sample sites. So two ways of really doing this is either setting up a particular site and having different numbers for each zone. So I've set this up here. I know that that 5 by 5 is 4. If I do a random number generator on the just on your phone, great things there, and it comes up 4. I know I'm sampling that area there. Or you could do something like this, where you set up 2 different areas. So you've got 1, 2, 3, 4, 5, A, B, C, D, E. Do a random number or a random number letter generator, I should say, on your phone, and where they actually intercept. So say I've got 3 and C, that is where you would sample. Now you need to think about, and this can take that bit of extra effort to set up those zones, but if you can, it does help remove bias from your sampling. Systematic sampling is a bit different to random sampling, and it still will have some aspects of being random, because that is quite important, that there needs to be some element of randomness to it to avoid bias. So systematic sampling is where samples from a larger population are selected according to a random starting point and fixed periodic sampling intervals, say every 10 meters, and this is determined beforehand. So let's just say here I've got this area, and I might set up my random number generator, and I've gone 1 to 100, and it comes up as 40. So I know that I'm starting at that 40 point, and from there I'm going to form a straight line, and every 10 meters I'm going to do some sampling. Alright, so you can see how this systematically samples along. This would be different to random sampling, which if you think random sampling, I've got, say, my numbers here, and I could be there, and then I might be here or here. This enables you to carefully go along a particular area, and it means that you are doing, and this is really good for, say, stratification or zonation, because you are going to be able to determine what happens across that zone. Where random, even though it's random, I might end up, say, doing there, and let's put another one here. And I actually don't get a great idea of this other area, or vice versa, depending on which way you're looking at it. So systematic sampling is a way that enables you to actually carefully notice any changes in zonation or stratification as you work across a zone. So that's what I've said here. Systematic sampling, this is one of the key things you should really get down and understand in systematic sampling, is it's useful for sampling study areas that include an environmental gradient. So if I had this area and I wanted to look at, well, what are the different changes in vegetation from the bottom here to the top? If I was to do random, well, I could end up just hitting all these parts at the top. Wherever I'm systematic, I know that I'm going to every, say, 50, 30, 10 meters, do a little sample there, and as a result, I'm going to get a good understanding of the vegetational changes as I move across those different strata as we look there. So you can see how that is useful. So an environmental gradient, which again, this is what we're talking about, exists whenever there is a gradual change in environmental conditions, so abiotic factors across a space. So things that you'll probably encounter being here in southeast Queensland or wherever you're most likely going to be doing coastal sand dunes. And you know that you're going to have that change from the high tide zone into the woodland, or it might be on the rocky shore. Again, you're going to notice a change, particularly on the water levels from that high tide zone to the low tide zone, okay, or high tide to low tide zone there. You'll notice those different changes. So environmental gradients can be related, and these are different things such as altitude, temperature, depth, ocean proximity, soil humidity, and species abundances will usually change along environmental gradients in a more or less predictive way. And systematic sampling enables you to actually notice those changes in species abundance better than random sampling if you do have some form of gradient. Now, if you are, for example, not wanting to notice, or your research question wasn't, is there a correlation or change as you move across the zone, and you just wanted to simply note and break your zones up into 3 clear zones and just see, is there any difference between the 3 zones? Well, in that case, random sampling might be a great idea, okay? Because you can just hit, I'm going to do this zone, collect some data within this area, this zone, collect some data, this zone, and see if there's any difference. If you're not looking for any sort of correlation or direct change in points as you move across. So it really comes down to what you're after and what you're looking for. So stratified sampling, and this is where we really go to in this particular section here, is many study areas are not uniform. Instead, they include a variety of distinct habitats, especially if the study site is large. And this is where stratified sampling is useful. If you have a variety of different habitats within a particular area and you're wanting to study them. So we'll see how this is a bit different to random and systematic. So in stratified sampling, the various habitats are sampled separately in proportion to their representation in total area. So for example, if I had random sampling and I was looking at this area and then I was looking at another area, well, I might be doing the exact same amount. Well, in stratified sampling, if this area is larger, well, I'll be doing a few more sampling points there. So one of the key things here in stratified sampling is the sampling is proportional to the size that's represented by that area. So stratified sampling divides the physical environment into smaller components for more accurate portrayal of your given area. So it does involve knowing how large the sampling area you're looking at is. So in stratified sampling, the area is divided into relatively similar subsections called strata. So if I was doing, say, vertical stratification as I'm looking in the rainforest, well, I could have my subterranean, my understory, midstory, and canopy. All right, and I've broken them into those different strata, as you can see there. And then I would sample within that strata based on the size that's given there. So it would be in proportion. So strata can be vertically arranged or horizontal, but it cannot overlap or leave gaps. So that's the key thing. If you're breaking your area down into different strata, it cannot overlap and you cannot leave gaps. So if we were looking at this example for rainforest, let's say you do go to a rainforest area and you're looking at some of that vertical zonation or stratification, you might divide it into the forest floor, the understory, the canopy layer, and the emergent layer. And you might be just doing some point sampling, looking up and seeing some different changes there. Or it might be horizontally stratified. So here I might break it into a couple of zones. You know, they've done these zones. For us, we'd probably go, okay, we're going to call this is our pioneer zone. This is going to be our colonizer zone, and this would be our woodland zone. And we break it up into those 3 areas. We would also then have to measure how large each area is. And if we were to look at just based on, let's say I'm setting up my pioneer zone, I should actually set it here, not to there, and then I've got this one, and then I've got this. Well, I might look at that and go, oh, well, my pioneer zone is actually a little larger, all right, as I set that up. So I'm going to need to do a few more quadrats. My woodland's a bit larger than my colonizer, so I'll do a few more here compared to that colonizer. So that's kind of the key idea. You break it into your different strata, okay, so let's say pioneer, colonizer, woodland, and then you do your sampling proportional to the size of that area. In that sampling, well, the things you might do that sampling as a random or systematic way, similar to what we've looked at previously when applied within each stratum. So I might do random sampling within each stratum, or I might do systematic sampling within each stratum. So samples should be taken from each stratum, as I said, in proportional to its representation in the total population. So if I was to sample these two different areas, let's just pretend that we've got, we'll call this the low tide zone, high tide zone on the rocky shore, and this is the area I have. Well, if I was going to do it in proportion, I can see that this makes up about 20% of the total area. So if I was doing 10 quadrats, I'd say I would do 2 in this area, and then I would do 8 quadrats in that area. So I've broken it down, and within each area, in this case, I've done it randomly, okay? So within here, I know I'm doing 2 quadrats, and I'd set that up randomly. In this area, I know I'm doing 8, and I'd set that up randomly, because I'm not looking necessarily, let's say I'm doing it this way for a particular gradient. Obviously, if I wanted to see the change from here to here, well, I would do it systematically, and I would do, again, in proportion as you move along that particular area. So if a habitat makes up 20% of the total area, it should be allocated only 20% of the total number of sampling points. That should be pretty clear and make sense to you there. So proportional sampling is an essential part of stratified sampling. If an ecosystem contained 30% eucalypt woodland and 70% grassland, the sampling fraction would need to represent this, i.e. 6 quadrats in the eucalypt and 14 quadrats in the grassland, if I was to do 20 quadrats in total. So if you were to look at these areas now, so we're trying to consider, I've got fernland, shrubland, sedgeland and closed heath. I want to sample and see if there's a difference in the abundance of, say, shrub species or grass species in those particular, or even just different organisms, whatever you might be looking at. So I've got my particular area that I'm going to examine, and I can do it in a couple of different ways, okay? So let's say I just randomly go to that area. So I randomly go, and I'm just going to sample all these different zones, and I put my quadrats down there. Well, if you look at this, you can see that the random sampling will simply ignore the relative size of each habitat. And it probably over-tests this sedgeland here, because there's quite a bit of testing going on in there. Very little testing on this fernland, which makes up a large amount. So it's not going to actually truly represent the size of each area. So while you're doing random sampling necessarily with stratified sampling, it can't be just, I go to the area, this huge different part with different habitats, and just randomly place down quadrats. I need to break it into the different strata. Okay, so I've got these 4 different ones here, and then I look at the relative proportion and I add that number of quadrats randomly in those areas. So if I was to do random sampling more effectively here, you can see that I've got 3 in the fernland because they make up the larger amount. I've got 2 in our shrubland, I've got 2 in our sedgeland, and I've got 1 in the closed heath. And you can see that now each area is actually sampled in a relative proportion to the size that it makes up. And I would do that randomly, okay? In this case, I'm not looking at a gradient or anything like that, so I don't need to do it systematically. I would choose a random method, but hopefully you can see the key difference here with stratified sampling compared to just a normal random sampling. Random sampling, I just come in, throw my quadrats randomly over the area, ignoring the relative size of those different areas, okay? However, in stratified sampling, I'm going to get a more accurate proportion of the habitat sampled with that stratified sampling technique because I actually take into account the relative size of each habitat, and then once I've got those strata and their sizes, I can look to either do random or systematic sampling within that. So in this case, I've just chosen to do random sampling, and you can see I've placed them down, but I should get a fairly accurate representation of that area with stratified sampling by doing that technique. And that's kind of the key idea just to set up. So we have 3 main sampling methods, okay, that we can look at: random, systematic, and stratified. In this unit, the key focus is stratified sampling. So it's basically breaking those different areas into the habitats or zones or strata, whatever word you really want to use. And then within that, once you've broken it down, you need to look at, do I just have random areas, okay, or is there a clear gradient? If there's a clear gradient between the strata or zones that you've looked at, well, you might do systematic sampling, where every so often you're putting down a quadrat after starting at a random point. If there's no real gradient you're looking at, say in this case, there's no real gradient, well, you do random sampling proportion to the size of each area in those particular habitats. Hopefully that makes sense, guys. Next lesson, we're going to start to look at sampling techniques as the next step in this process.